Today, I will be talking about using CRISPR engineered flies to study VCP disease mutations. So what is VCP? VCP is a AAA ATPase, and it consists of an N domain, a D1 ATPase domain, and a D2 ATPase domain. The N domain is a cofactor binding domain, whereas the D1 ATPase domain plays a major role in VCP hexamerization. And finally, the D2 ATPase domain is responsible for ATP hydrolysis. So what is the role of VCP? So VCP has many roles within the cell, and so just to name a few as seen in this picture would include autophagy, stress granule clearance, endosomal sorting, and ER-associated degradation. VCP is a causative gene for a broad spectrum of degenerative disorders. So there's currently more than 30 known disease-related mutations. So normally the VCP gene encodes for an ATPase enzyme which is utilized in the cell for a variety of functions that includes cell division, fusing membranes, apoptosis, and repairing DNA damage. However, in degenerative disorders, the mutant VCP proteins will co-localize protein aggregates in neurons, which is thought to be a major factor in driving neurodegeneration. So some of the neurodegenerative diseases that are associated with mutants in VCP include IBPFD, ALS, spastic paraplegia, and muscular dystrophy. And one of the major symptoms within neurodegenerative disorders is muscle wasting, which is seen in this figure. And I also want to note that all of the mutations of VCP are autosomal dominant, meaning that only one copy of the allele is required to cause the disease phenotype. Although VCP causes a lot of neurodegenerative disorders, the main disorder that I am focused with is IBM. So IBM-PFD stands for inclusion body myopathy with Paget's disease of the bone and frontal temporal dementia. So IBM-PFD is an autosomal dominant progressive disorder that occurs around midlife, so roughly 42 years old. This disorder varies in its pathology and clinical penetrance between patients. So if we break down IBM-PFD, we have three different types of symptoms. So the first symptom is inclusion body myopathy, and this symptom causes muscle weakness around the skeletal muscles and affects roughly 90% of individuals suffering from IBM-PFD. The next symptom is Paget's disease of the bones, and this affects roughly 50% of individuals suffering from IBM-PFD. And it causes affected bone to become fragile and misshapen, and it commonly occurs in the pelvis, skull, spine, and legs as seen in this picture. And then finally, the last major symptom associated with this disorder is frontal temporal dementia, which affects roughly 33% of individuals suffering from IBM-PFD. So animal models are very useful for studying human diseases. In particular, flies can be genetically engineered using technology such as CRISPR to express human disease mutations. This allows us to study the pathogenesis in a model organism that has a relatively short life cycle. And we hope that by studying the disease mechanisms in fly models, we can discover new strategies to treat the disease in humans. Using CRISPR to knock in mutant disease genes in flies. So this explains how we use CRISPR to insert VCP disease mutations. So the first thing you do is you have a gene, and for this example we have VCP. Then the CRISPR-Cas9 will cut this gene out, and then with the use of a donor template plasmid that's carrying the VCP mutant, it'll insert it in through the natural homology directed repair. Our animal model, which is Drosophila, will now have a VCP mutant inserted into its DNA. So Drosophila contain a VCP homolog, which is known as TER94, and TER94 shares 93% similarity with human VCP. So by using CRISPR gene editing, we were able to knock in 10 of the most prominent and severe VCP mutations at the endogenous locus. Using CRISPR to generate endogenous VCP disease models. So like I said previously, we looked at the 10 most prominent and severe VCP mutations. So the ones that we ended up using for our model were P134L, R152C, R152H, A154R, R156C, R188Q, and L195W, and these are all involved with the CDC48 homology, and they're used for substrate / cofactor binding. And then we also looked at T259A and A436S, which are involved with the D1 domain, and they use the hexamerization and ATPase activity. So the major goal of this research was to understand how each individual mutation affects VCP function, which could inform new strategies for personalized treatments in the future. So the first thing we looked at was longevity, and we found that VCP mutants have a minimal effect on lifespan. So these are 4 of the mutations that actually had a significant effect on lifespan. So you have R152C, T259A, R152H, and L195W. However, the rest of the mutants, such as R156C, R188Q, P134L, and A154R, and A436S, were mutants that did not show any significance in regards to longevity. So next we did a negative geotaxic assay to examine motility. And so the negative geotaxic assay is used to demonstrate muscle atrophy. So flies naturally want to go towards the top of the vial. So to test their mobility, I would mark the food from the food to the top of the vial 3 centimeters and allow the flies roughly 10 seconds to get to the top of the vial. So whenever we look at the wild type, what you see is as soon as I start the timer, all the flies go straight up to the top, and then you can see they don't have any issues with this. But whenever we look at the mutant, we see something completely different. So as you can see with this mutant L195W, they have a really hard time being able to get up to the top of the vial. And although some of them do end up crossing the line, you can see a lot of them are still struggling. They're kind of almost just stuck to the side of the vial. So whenever we compare this, the wild type versus L195W, you see that although the wild type does drop off a little bit, even comparing it to the mutant, around only 80% at week 1 are even able to climb up to the top. And then when comparing week 3, then we have 40% are only able to climb to the top. So it's a pretty drastic decline between the wild type and this mutant. And so therefore this indicates that our fly mutants are mimicking the phenotypes that are observed in human patients with VCP diseases. So VCP mutants display progressive motor impairment mimicking human disease symptoms. So in this picture, this is week 3, and so this is like I showed previously between the wild type and L195W, there was a drastic decline. And so as we can see here, we have a lot of the mutants were actually extremely significant. So R152C, A154R, R156C, L195W, R188Q, and A436S all showed significant decline in their ability to climb to the top of the vial versus the wild type. Next, we did an aversive phototactic suppression assay. And so for the first step we do is to collect flies that are phototaxic. So the way that we do this is we use a T maze, which consists of a light chamber and a dark chamber. So the vial that I'm pointing at represents the dark chamber, and this vial represents the light chamber. So what I'll do is I'll insert the dark chamber into the T maze and shine the light into the light chamber. And as you can see, the flies naturally want to go towards the light. So then I will collect these flies, and these will be termed the phototaxic flies. So then I will do training periods, and so the training periods consisted of 12 trials, which each lasted roughly 60 seconds. So in the training period, I used an aversive odor. So since flies naturally are attracted to the light, when they're paired with this aversive odor, what we see is that with the wild type, over the course of the training period, they actually remember that the aversive odor is present, and they actually stay in the dark chamber and don't migrate to the light chamber. And so after we do the training periods, then we do trial runs. So we do 5 trial runs immediately after the training period. And then we analyze the trial runs to see the memory impairment. So after analyzing the data with the aversive phototactic suppression assay, we identify that VCP mutants exhibit memory impairments. So by comparing to the wild type, we find that a number of VCP mutations are actually significant in that they impair memory function. So some of the mutants include P134L, R152H, A154R, R156C, L195W, and A436S. So the conclusions of this project are that some VCP mutants have a negative effect on longevity, VCP mutants have a progressive negative effect on mobility, and some VCP mutants display memory impairments. So for my acknowledgements, I would like to thank the Discover Undergraduate Research Program at Louisiana State University who funded this research, and I would also like to thank the College of Science and all of the members from Johnson's lab. Thank you.